Okay, we see you, but we do not hear you yet.
Yep, here I am.
Okay, now we also hear you. We are listening.
All right, fantastic. Hello, everyone. Good evening. My name is Margie Christ. I'm a PhD student at the University of Western Ontario in London, Canada. And I'm also thrilled to be a part of the wonderful intern team at the Quantum Biology Laboratory at Howard University. So I hope that no one's tired of slime molds at this point in the day. If you heard Dr. Kurian or Dr. Bajpai earlier, you've heard already a little bit about our slime mold work. So the aspect of the project that I have been focused on is looking at solutions to the traveling salesperson problem with slime mold inspired algorithms. So as you may know, the TSP is a root finding problem that is NP hard, meaning we don't have the ability to solve an arbitrary TSP in polynomial time. There are methods and approximations to find reasonably good solutions to various types of TSPs, but we don't have an overall solution that is feasible for any problem size or any problem configuration. But one interesting thing that can be observed in the slime mold behavior is that Fusarium polycephalum, when placed on a nutrient-rich agar plate containing a multi-lane chip, can, in response to optical feedback from a modified Hopfield neural network, actually solve the traveling salesperson problem. So these results were found experimentally. A team in 2018 did this with the actual Fusarium. You can see in figures 1 and 2. In figure 1, a diagram of the experimental setup where the slime mold lays on the chip, which is on the plate, and is illuminated by a projector. And then the data is processed at a computer. Figure 2 shows a solution to an 8-city TSP, where you can see the blue and the green are where the slime mold has chosen to go. So it selected 8 of its possible options to determine what order it's visiting the different 8 locations. So after observing this experimental behavior, the first team, Zhu et al., wrote a computer program to mimic Fusarium's behavior. And then subsequently, a second team of Miyajima et al. Provided modifications to improve this computer algorithm's performance, namely by reducing the number of iterations required to reach a solution. So what we looked at specifically regarding these two codes is both codes require the addition of a small amount of computational noise in order to successfully reach a solution. That's added as a uniform random number distribution for the first code, or a Gaussian or normal distribution in the case of the modified or the second version of the code. We were hoping to minimize the performance metrics of solution length and iteration count. And to do this, we varied the noise's magnitude and the noise profile, switching them between the two codes. And then we also changed a couple of code parameters to hopefully better mimic Fusarium's polycephalum's behavior. There are small influx and outflux fluctuations that are expressed through two variables, one for expansion and one for contraction. And so we examined the effect of changing the ratio of these quantities. If you look at the results in the bottom left regarding our first goal, the original code from Zhu et al. Tends to minimize root length. And the second code, the update, minimizes iteration count. However, the differences in solution length are typically within one standard deviation of each other. It's not a huge difference, but the differences in iteration count are typically pretty big. You can see in figure 4 that there's almost an order of magnitude difference between iteration count for the first version of the code versus the second version of the code. And so when we compare the logarithmic product of our two success metrics, We see that the updated code with the normal noise profile presents the most computationally efficient method without compromising solution quality. And regarding our second goal, varying these influx outflux parameters, again, the solution length does not vary significantly when we change these quantities, but the iteration count in both cases for both codes is minimized when the ratio of expansion to contraction is less than 0.5. So you can see in figure 6 that in the case of the first code, our best results are when the expansion is very small, and with our expansion over contraction ratio being at 0.1. And in figure 7, you can see that similarly, but not exactly the same, our best results come when our ratio of expansion over contraction is at 0.25. We also have some connections to other work going on in the QBL. So methods such as the Lynn-Kernighan-Helsgaun algorithm, which is our best TSP heuristic algorithm, do provide high quality solutions, often within less than 1% of the optimal Tor length for the TSP. But there are also other options that can be explored for finding solutions to the traveling salesperson problem. One of these is an unstructured database search, which then connects us to Grover's algorithm, the well-known quantum algorithm for unstructured database search. And it's interesting to note that this algorithm can be implemented upon any system that supports superposition, such as a system of coupled harmonic oscillators. And this connects back to biosystems by mention of a model of the algorithm applied to the problem of DNA search repair, where an unpaired nucleotide base must select its correct partner from an equal superposition, essentially, of the 4 possible nucleotide bases. It needs to find the solution to its database search in order to partner correctly. And we're hopeful that coupled oscillator implementations of Grover's algorithm may prove useful in understanding functionality and efficiency of a wide variety of questions around how DNA functions. So in summary, we are looking at how bio-inspired computing algorithms can help us solve computationally difficult problems. There's also a future improvement that we're hoping to make by changing the noise profile from white noise, which has a constant power spectral density, to pink noise, which has a PSD of 1 over the frequency, as pink noise is commonly found in biological systems, and we're hopeful that will provide a better computational approximation of the slime mold's behavior. So I hope this small snapshot of some sort of different topics in quantum biology was interesting. I think this field is fascinating, and I'm so excited to be working on such cool and interesting projects with the QBL. Thank you so much for having me, and I welcome any questions.